Bueno, buenas tardes. ¿Me oyen? ¿Funciona esto? Ok. Bueno, buenas tardes a todos. Yo creo que vamos a empezar. Bueno, pues hoy es la primera sesión de nuestra nueva periodo de Frontiers in Genomics. Y hoy tenemos el gusto de tener al doctor Ashenberg con nosotros. Él trabaja en el Broad Institute en Boston. Y debo decirles que yo entré en contacto con ellos porque el año pasado en un congreso en Boston justamente tuve la oportunidad de escuchar a una persona del mismo grupo de él trabajando sobre el concepto de un atlas de células en el Broad Institute. Y me pareció que era muy interesante para el curso de Frontiers in Genomics, obviamente que era un must. Y entonces empecé a escribirles y finalmente el doctor Ashenberg aceptó la invitación que les hicimos. Y les voy a decir que él es una persona que ha hecho es graduado de MIT, es biólogo computólogo, y después hizo un doctorado también en MIT y hizo un postdoctorado en el Fred Hutchinson en Seattle y desde hace unos años trabaja en el Broad Institute justamente en el grupo que está desarrollando esta maravillosa idea de hacer un análisis a nivel de células únicas de las células de un organismo vivo, ¿no? Entonces, también quisiera, ya que estoy haciendo esto y porque siempre se me olvida, es agradecer a las personas que están en línea con nosotros. Hoy sabemos que están con nosotros el Centro de Biotecnología Genómica de Reynosa y aquellos otros que están con nosotros también les digo que para hacer el chat en vivo, ahora seguimos la misma liga que antes, pero esto los va a llevar a un canal de YouTube y esperamos que la conexión sea más fácil que lo que era antes, porque antes era realmente complejo poder ver en línea y en tiempo real las conferencias. Entonces, la liga es www.lsg.unam.mx/frontiers/live. El título de la conferencia es Developing a Single Cell Genomics Toolbox for Human Tumors. Or I would like to thank you very much for coming. And I think for the students it was also a very nice experience and it was really a pleasure to interact with you.
He's working? Okay, maybe I just wasn't, there we go. Okay, great. So thanks for the introduction and thanks to Yvonne and Uzil and the students. It's been really fun visiting here and learning about what's going on and kind of talking about some of this research. So today I'm going to talk to you about the development of different single-cell genomics approaches and how we're using that in studying human tumors. So we know there's this incredible diversity in cells as we look across the human body in both healthy tissues and diseased tissues. Let's see. So here we're looking at different images throughout the human body. Here, for instance, we're looking at blood vessels, and monocytes can migrate from the blood vessel into the tissue and develop into macrophages. Here we're looking at small intestine. We see this beautiful structure of these crypts, where at the base there's these intestinal stem cells, and as you go further along the crypt, there's this differentiation process into the different epithelial cells. And then there's also very interesting differences in cells as you go into disease tissue. So as you're looking at diseased liver or breast cancer. And my big focus is taking single cell tools and applying them to understanding cancer and tumors. So tumors are almost like one of the ideal systems in which you would want to apply single cell tools because there's this incredible diversity in both the malignant cells within a patient, and there's these very important interactions that take place between the malignant cells and the stromal cells, so like endothelial cells, fibroblasts, the immune cells, so with branches of both the innate and adaptive immune system. And there's all these interactions that are taking place in the tumor. And we know that the malignant cells themselves can have genetic diversity. There can be different clones of them. We know that the T cells and the B cells can be parts of clonotypes. So there's this very interesting and important set of interactions that are taking place. We want to be able to understand what every cell is doing. We also want to understand what happens when we perturb the system, when we treat a patient with a drug, or how a cancer changes from a primary cancer to a metastatic cancer. So looking at how things are changing over time. And so today I'm going to talk to you first about some studies we've done with tumors and their microenvironments. First talking about tumors, then immune cells, then combining them together into a study. And then in the second half of the talk, I'm going to talk to you more about methodological developments that we think are going to help the field as a whole. Okay, so the first half of this talk, there's sort of three little sections here. First, I'm going to talk to you about work we've been doing in glioblastoma. Then work on profiling the healthy immune system, and then a piece of work that kind of looks at both the malignant cells and the immune cells together and how they talk to one another and how they influence one another. So this is a study from 5 or 6 years ago now, and it's a study in glioblastoma, which is an adult brain tumor, and this is one of the most common brain tumors and also one of the most aggressive and very high mortality. And what we did in this study was we took glioblastoma samples from 5 patients. So these were resections that were taken out during a surgery. And we collected the tumor and dissociated using enzymes and isolated single tumor cells. And then we wanted to know what are the genetic programs that are on in these tumor cells from these patients. And so 5 or 6 years ago, one of the most popular techniques for doing single cell sequencing and looking at gene expression was to sort single cells into 96 well plates and then prepare the transcriptomes of each of those cells in a 96 well plate. And how this works is we have the mRNA from a single cell that's been lysed. We bind it with an oligo DT primer that binds the poly A tail and do reverse transcription to get a first strand cDNA product. And then with some standard sort of library prep techniques, we can make a library that can be sequenced on a next generation sequencing device. Okay, so once you have done this and you have got these transcriptomes for every single cell, you can think now of every single cell having measurements for 20,000 different genes. And if you make that into a matrix, now you have genes as rows and cells as columns. And here we measured 430 different cells from these 5 glioblastoma patients. And then we go forth and do some analysis, computational analysis, which is what I focus on. And so the first step in any analysis is quality control. So we want to find cells that are high quality. So often in single cell analysis, collecting the tissue, dissociating it, getting the single cells, it's very stressful for the cells. So we don't expect that every single sort of experiment will work very well. So there's always some quality control we do on the cells themselves. Then we carry out a normalization step, which is essentially every single cell gets sequenced to a different depth. And so there's all these little technical differences between the cells. And so we want to normalize across the technical differences before we start comparing cells to one another. After that, we do what's called feature selection, which generally means that we only look at the genes in the cells that are variable. So if a gene is uniformly expressed across all the cells, we throw that gene out because it's not going to tell us anything about the biological variation in our data set. And conversely, genes that are very highly variable are probably going to be the genes that are going to be most interesting because they're maybe highly expressed in one group of cells and more lowly expressed in other groups of cells. So this removes a lot of the genes from the data. So remember, we start with 20,000 genes, and after we do this sort of feature selection, we often have 2,000 to 3,000 variable genes remaining. That's still a lot of genes. And so the next step we often do is a dimensionality reduction step, which means we're going from having 2,000 or 3,000 genes to some reduced set. And the reason we do the dimensionality reduction is for many reasons, but maybe the main reason is that the genes in any cell across your body, they don't vary independently of one another. So genes are often turned on or off together. So you don't actually need to measure the expression of all the genes in a cell to know what all the genes are doing, because the expression of one gene is correlated with the expression of another. So if you know one gene's expression, you know the other. So a common dimensionality reduction approach we use is principal components analysis, which is identifying linear combinations of genes that maximally explain the variance in your data set. And those principal components often have biological meaning, like the first principal component may separate immune cells from non-immune cells. Okay, so once we've done that dimensional reduction, we've gone from having 2,000 or 3,000 genes to often 50 to 100 principal components. So now the data is on a much lower dimensionality, and we can start doing things like clustering, where we get the distance between every single pair of cells. We build a graph saying which cells are next to one another, and then from that graph, we assign clusters to each cell, so which cells belong together. And ultimately, what we find in this clustering is that cells cluster together if they have similar gene expression profiles. And now we want to interpret what those clusters mean or do not mean. Okay. So we do this approach on the glioblastoma cells, these 430 glioblastoma cells, and we visualize that high dimensional data in a 2D plot. And so here are the 430 glioblastoma cells, and they're colored by which patient they came from. And what you can see is that the cells actually cluster separately from one another, meaning there aren't huge commonalities between the cancers of these various patients. And that is interesting, but maybe not surprising, because we know that cancers are genetically heterogeneous, and one individual's cancer is probably going to be different from another because there's different genetic drivers. And so, but this is very interesting because really at the single cell level, we can observe both the diversity within the same cancer. So these are cells all from the same patient, but they're clearly not really all the same. But these cells are also different across patients in their gene expression profiles. So we see this really amazing heterogeneity when we look at an individual tumor and we look at the cells within that tumor. Furthermore, we can rely on other work that's been done in the past. So there was a big effort called the Cancer Genome Atlas, which was profiling many different cancers across adults and doing things like bulk RNA sequencing. And from those efforts, people identified subtypes of cancer, so subtypes of breast cancer, subtypes of glioblastoma. So there were these 4 sort of subtypes of glioblastoma that were proposed, called proneural, neural, classical, and mesenchymal. And these are just different sets of genes that are highly expressed within those subtypes of glioblastoma. And now you can take those signatures, and within each patient, you can score every single cancer cell for the expression of those signatures. And so what you see is that within a given patient, it's not as if all the cells cluster around being like high in the proneural signature or high in the classical. What you see is that usually the cells are spread between multiple signatures, suggesting that there's actually within a patient, there's high subtype diversity. So it's not like a single patient has one subtype. It's often maybe 2 or 3 subtypes, and there's this sort of continual differences between those subtypes. It's not as discrete as maybe we would like it to be in terms of thinking about treatment. Like if there was a very good treatment for proneural cells, that's great, but it still leaves out the other cell states that are found in this patient's tumor. So this was very kind of interesting and sort of foundational study in single cell for applying to tumors, because you're seeing this really high degree of heterogeneity both within patient samples and across individual cancer samples. And it's very informative when it comes to thinking about treatment, because if each cancer is a bit different or very different, that means that we need to think about which treatments are appropriate under different contexts like these. Okay, so that was a study that was done 5 or 6 years ago, and since then our ability to do single cell genomics has really taken off. So here's a chart showing different single cell studies over the years, starting in 2009 from the first study, and on the x-axis the time of the study, and on the y-axis is the number of cells that the study was able to profile in at least the transcriptional level. And what you can see is that it's really taken off, and this is a log scale, so it's really exponential growth, where we've gone from 5 years ago being able to do about 100 cells to now being able to do around 1 million cells without too much work. So that's a really unbelievable amount of growth. Like, it's hard to compare this to anything else that I've seen in biology, other than maybe things in like the tech world with processing speeds and things of that sort. So it's very exciting because now we can profile many, many, many cells, and it's getting cheaper to do so. And that's become doable because of just improvements in technology and in sequencing. The main improvement in technology has been moving from doing things manually with pipetting to microfluidics, where we can control the flow of many cells and droplets and things of that sort. And I'll give you an example of that shortly. So that was looking at malignant cells. In parallel, we've been looking also at immune cells to try to understand what does the healthy immune system look like. And so in this study, we collected tissues from healthy adults from both peripheral blood, bone marrow, and cord blood, which comes from the umbilical cord. Which comes from the umbilical cord. And these are the number of cells we profiled from each of the tissues, and the number of donors, and the sex differences there. And what you can see is here we're looking at like hundreds of thousands of cells. So we did close to 2 million cells. And we did this about 2 years ago now. And so really an incredible number of cells being profiled, and it lets us look at both rare cell types or rare cell states that maybe you need to sample a lot to observe. It also lets us look at variation across individuals because we've sampled a few individuals here. So this is to analyze this data with 1.7 million cells, it quickly becomes very computationally challenging. And so we developed with Bo Lee, who was a former postdoc in the group, we developed a cloud-based pipeline to do a lot of the analysis. And again, the reason this is so hard is because you're starting with a matrix that has 20,000 genes, 20,000 rows, and 1.7 million columns, one for each cell. So if you've ever used R or Python and try to load such a matrix into memory, you're going to run into problems. So you really need new ways of doing some of the analysis. And so we developed this cloud-based pipeline that runs on the Google Cloud, but it can also run on Amazon and whatnot. And this really sped things up quite a bit and made it, made it possible for us to process data sets within a few hours rather than like waiting weeks or months. Right, and so we're doing everything in the cloud, little cloud image here. And so all the analysis steps we also try to make as parallelized as possible, so you could split it across multiple processors. So we have our quality control, variable gene selection, dimensionality reduction, clustering and visualization, all sort of sped up as much as possible. And so this is what we were using to do this analysis. And now these tools are publicly available. It's on bioRxiv, and all the code and instructions for doing this yourself are there. So you can actually run single cell analysis in the cloud, and it's all Dockerized and pretty, hopefully straightforward to use. But we kind of put this out there. And here is one of the results. So this is 300,000 peripheral blood mononuclear cells, where we are visualizing them in 2 dimensions and coloring them by the assigned cluster, where we further annotated the clusters based on differentially expressed genes. And based on those differentially expressed genes, we can know if something's a B cell or a T cell and so forth. And what you see in this clustering is all the major blood cell types you would expect, the myeloid cells and the lymphoid cells. So we see B cells, we see a big cluster of T cells, we see the NK cells over here, and then we also see myeloid cells, like different monocyte populations, different dendritic cell populations, and so forth. And then you can look at this at increasing resolution and start to look at T cell states and things like that. We can also look at how this varies across individuals. So remember, this data set came from multiple individuals. So in this plot here, we just color the cells based on which individual they came from. And what you can see is that within every cluster, there's sort of this rainbow of colors, meaning that every cluster of cells is represented by multiple individuals, which is kind of what we expect to see, that everybody has more or less T cells, B cells, monocytes, and so forth. But we're looking further into this data set to see how expression differences might correlate to differences in genetics, so identifying things like quantitative trait loci and things like that. But one thing that was good was we didn't see a strong batch effect where, for instance, we only see a cell type in one individual, but not in all the other individuals. So we did this for, like I said, 3 tissues: bone marrow, cord blood, and peripheral blood. And here's sort of the 30,000-foot view of what those data sets look like. And this research was part of the Human Cell Atlas, which is trying to profile healthy tissues throughout the human body. And we put all this data online like maybe a year and a half ago before even having made any publications on it. And this is kind of a unique model. You know, this study costs like millions of dollars, but we actually haven't written a paper about it. We just put the data up so other people in the community could use it as quickly as possible, because as you know, the time to publication is quite long, and we wanted this, to be useful for people. And actually, there's been dozens of papers written already using these data sets, both using it to understand biology and also developing new analytical approaches, because the data set's so big that it really stresses any algorithm that you've developed, because it's just you need to be very efficient to process something so large. Okay. In this final study I'll go through, we were combining both looking at the malignant cells and the immune cells. And the question was, why do some melanoma patients respond to immunotherapy and some do not? So we know that the success of immunotherapy and whether a patient responds to it and survives longer post-drug treatment is often related to whether the tumor is infiltrated by T cells. So in immunotherapy, you're often trying to modulate the activity of T cells so they go and kill the cancer. But if there are no T cells in the cancer to begin with, then an immunotherapy probably will not have the effect you want. So the question we were asking was, what is it in the tumor that determines whether there's a high level or low level of T cells infiltrating into that site? And so the approach we took was to look at 31 different patients, untreated patients, and ones who were resistant to the immunotherapy. And we did single-cell RNA sequencing on those tumors. And we looked at both the malignant cells and the stromal cells and the immune cells. And in parallel, we also did bulk RNA sequencing, which is much easier to do than single-cell RNA sequencing. And we had a data set for that of close to 500 patients. So two different data sets here. And here is the clustering for the immune and stromal cells. And you see some large clusters here. I'll just tell you that these are the immune cells here and stromal cells here. And then here's the clustering of the malignant cells. And for the malignant cells, you see many, many different clusters. And this is very similar to what you saw with glioblastoma, where each patient's tumor cells clustered separately. And so we see that again in melanoma. And actually, we see this in pretty much every tumor that we profile by single cell, that patients' tumors are very different from one another, kind of underscoring the importance of thinking about heterogeneity when dealing with cancer. Okay. So then what we did was we took that single-cell RNA sequencing data and we defined signatures for the malignant cells and the T cells. And by signatures, I just mean genes that are highly expressed in malignant cells or T cells, but not highly expressed in the other cells. And then we took those T cell signatures and we used them to estimate the level of T cell infiltration in the bulk data. So we had this cohort of 500 patients where we've done bulk RNA sequencing, and we asked, what's the expression level of the T cell genes that we've derived from the single cell RNA sequencing? What's the expression level of those in the bulk data? And so the point of this is to say, is there high T cell infiltration or low T cell infiltration in these 500 tumors? And then we have a very big data set now where we have 500 patients with data, and we want to ask, what are genes that explain high levels of T cell infiltration or low levels of T cell infiltration? And so all we did simply was look for malignant genes whose expression was correlated or anti-correlated with T cell infiltration. And then that became our signature for T cell exclusion. So this is a signature of genes that we think explains or is correlated with many T cells being at the site of a tumor or few T cells being at the site of the tumor. And here are the genes that are in that signature. So these are genes that are highly expressed in the exclusion signature, and these are genes that are lowly expressed in the T cell exclusion signature. And you don't need to look at all these genes, but I'll highlight a few. There's CDK4 and SOX4, so these are important in cell cycle, and is an important transcription factor, so that's up. So cycling is up in malignant cells that are excluding T cells. Genes that are down, well, you see a lot of HLA genes that are down in B2M. So these are genes that are important in antigen presentation. So these are actually lowly expressed in the tumor cells, and that can be a way for the tumor to avoid being recognized by the T cells. So it's sort of immune evasion strategy, which is pretty common across many tumors. So if the tumor isn't presenting many non-self antigens, the T cells can't recognize them and kill them. So a lot of these genes made sense in terms of genes that you would expect a tumor to use in terms of evading being recognized by the immune system. We also did some validation of this using immunofluorescence. And so here is a section of melanoma, and in red and green are cells with high MHC class 1. So that was these HLA genes that I was talking about here, with high MHC class 1, which is associated with antigen presentation. And then in blue and cyan are T cells. And so what you can see is you see these large sort of islands of malignant cells where most of the ones in the interior are green, so they have low MHC class 1. And in that green area, you do not see many T cells. However, if you look at the borders of these sort of tumor islands, you see a lot of malignant cells that have high MHC class 1. So these are the ones that are presenting antigen to the immune system. And then around there, you see large levels of T cells. So this was sort of this was a validation that this genes we were identifying actually correlated with levels of T cell infiltration, at least at the spatial level. And then finally, what we did was asked whether this exclusion signature, these genes that are associated with T cell exclusion from a tumor, whether they can predict whether patients will respond to immunotherapy. So we have roughly 100 patients here, and biopsies were taken before they were treated. And then we did bulk RNA sequencing on those biopsies and scored them for the expression of this exclusion signature. And this is time post biopsy and treatment from 0 to 1 year. And this is progression-free survival. So this is a survival curve. And what you can see is that if you divide the melanoma patients by high, moderate, and low expression of that exclusion signature, you can actually predict progression-free survival on how well they are responding to immunotherapy. So patients that had low expression of this exclusion signature that prevents T cells from going to the tumor, they actually are doing better post-immunotherapy. And patients that have high expression of that signature to begin with, where they have many genes that prevent the T cells from actually infiltrating, many genes being expressed, they have low survival. So this was very strong validation that the signature we found from this single cell and bulk RNA sequencing study actually has predictive value in saying whether a melanoma patient will respond or not respond to immunotherapy. So this is pretty important because immunotherapy, as many of you know, is a very often very toxic treatment to give a patient. And so the more you can say whether or not someone will respond to a treatment, the better patient care you can give. So you can avoid treating patients with something that's not going to work and will probably be uncomfortable or make their lives more difficult as they're undergoing treatment. So this was very nice. And then sort of the next follow-up steps are asking, how can you take a tumor that has high expression of this exclusion signature, of these genes that prevent T cells from coming in, how can you take those cells and drive them to a state where now T cells can come in? So how can you reduce that exclusion signature? So for that, we've been doing a lot of drug screening, and there's some interesting results that we found doing that approach. But this sort of shows you how single cell RNA sequencing can be used to understand sort of the interplay between the immune cells and the malignant cells and hopefully benefit patients at some point. Okay. So just to summarize the sort of first half of the talk here, I talked to you about malignant cells and glioblastoma. This was one of our earlier studies. And then sort of the technological improvements and computational improvements we made to look at things in more high throughput. And finally, this study with melanoma and looking at what differentiates people who respond and do not respond to immunotherapy. Okay. So for the second half of the talk, I'm going to focus on what I've been doing for the past year, or maybe 2 years now, which is coming up with more generalized experimental and computational approaches for profiling human tumors. And so I'll explain to you why we need that. So I'm going to talk to you about optimizing single-cell transcriptomic approaches, single-nucleus transcriptomic approaches, and I'll explain what that is in a second, and then how we compare data from both of those approaches. So we wanted to develop improved experimental and computational methods for analyzing tumors. So most studies out there in the literature, they look at one tumor type and they develop a method to dissociate those cells and study them. And it's often a very specific method, but if you're looking at a new tumor type, there's often no guidelines for how you should approach that. And every tumor is going to be slightly different. So to get it like a general toolbox for analyzing tumors, we wanted to look at a very big variety of tumors. So we looked at many different cancer types, so non-small cell lung cancer, neuroblastoma, metastatic breast cancer, and so forth. These tumor types vary in their cell of origin, so some are epithelial, some are neuroendocrine. These tumor types vary in being solid or non-solid. They vary in whether they're pediatric or adult. So we tried to span as many different features of tumors as we could when we were developing this toolbox. And this is the number of patients we looked at and just the different samples that we were processing. And so why are we doing this? Well, like I said, every tumor type has its own challenges. So one of the main things is cell dissociation. So every tumor has a different set of extracellular matrix genes being turned on, and it's occurring at different tissue site. So looking at glioblastoma and is going to be different from looking at a breast cancer that's metastasized to the liver. Just how you break apart those cells are completely different considerations. So that's one complicating factor. It's often hard to dissociate cells, and it's also very time sensitive. So this is a very tricky procedure, because this isn't just something where you have a mouse and you sacrifice and you can do things immediately. This is something where it's coming out of a patient in an operating room, and you have a surgeon and you have a pathologist, and there's all these treatments that need to be done to the patient that are more important than your research. So getting that sample in a way where it's not sitting in the operating room for hours, or that it's going through a technical process that's taking a long time, is really important, because we know as soon as we start dissociating cells in a tissue, there are gene expression changes happening, and we don't want to be studying artifacts, things that aren't really reflecting the actual biology of the tumor. So time is really important when doing these types of things. And also, we need methods that account for the fact that there are different sample sizes. So a resection is when you take out like an entire tumor, and sometimes these can be very large. A biopsy sometimes can be done with just a fine needle. So there's very big differences in the sample size you're dealing with. And why does the size matter? Well, the more cells you have to work with, generally the easier things are. So these were features that the experimental team that I was working with was trying to sort of get past, or challenges they were trying to get past. And here's sort of a roadmap of a set of suggestions and decisions that you have to make as you go from collecting the sample to dissociating it and cleaning it up and preparing it for single-cell RNA sequencing. It's not showing up here, but the point is you can kind of see there's a bit of a decision tree in terms of what you should do, what you need to consider at every single step. And this can be applied to any tumor that you're working on. And to sort of validate this approach, what we did was very careful comparisons where we would take the same tumor and divide it into chunks and then process those chunks using different approaches, and then compare what does the quality of the data look like. And so what we were comparing was cell quality, the number of recovered versus expected single cells, and the cellular composition and how well that reflected what we expected to see in the tumor. So here we used, rather than sorting cells into plates, we used microfluidics and droplet-based approaches, and we used a solution from a company called 10x, which you might have heard of, but it's the sort of most popular approach right now to do single-cell transcriptomics. It's popular because it works well, and it's a standard solution where you can buy an instrument and follow a set of instructions and get thousands of cells pretty easily to work with. And how it works is you take your cells and you flow them through a microfluidic device. The cells get captured in a droplet, and in that droplet there's a bead that has DNA oligonucleotides on them. And every single bead has a different set of barcodes, a cell barcode. So every bead has a unique cell barcode, and that cell barcode gets introduced into the mRNAs of the cell during the reverse transcription step. So all the mRNAs from each of your single cell get a different cell barcode, and now once they've been barcoded, you know that they came from one cell or another, and you can mix all the mRNAs or cDNAs together and process everything in parallel. So that's very nice, and that's kind of a common approach that's used in the field now in other technologies other than 10x. And so one of the first things we did was we had these 3 protocols that we were comparing for lung, non-small cell lung cancer. And we first look at the sequencing data and we ask, what fraction of the sequencing reads are aligning to the genome, what fraction are aligning to the transcriptome. So we would like most of the sequencing data to align to the genes we know about. Sometimes things will align to regions between genes, and we think that's probably contamination from DNA that's come through. We also look at how well we can detect the different transcripts. So here is for every single protocol, how many sequencing reads per cell, how many genes we detected per cell, and fraction of mitochondrial genes being expressed. So number of genes detected per cell is a common metric we use to look at quality. So the more genes we detect per cell, generally we think the better job we did. Mitochondrial gene expression, why do we look at that? When cells are damaged during the single cell dissociation process, there can be holes in the cell membrane and maybe your mRNAs leak out from the cytoplasm, but the genes being expressed from the mitochondria are protected by that organelle. So often cells that have high mitochondrial gene expression are those cells that have lost a lot of their cytoplasmic mRNAs. So we often filter out cells that have high mitochondrial genes being expressed. And so we have these 3 protocols and we can compare them. And so, for instance, we see that across the 3 protocols, there's similar number of sequencing reads per cell, but one protocol, C4, has a higher number of genes being detected per cell. And that's good because we think that protocol gives us more biological signal that we can work with. So the more genes we can quantify, the more differences we can detect. Okay. Another thing we do is we do filtering for doublets. So to remind you, every cell is being encapsulated in a droplet with a barcoded bead. However, this is sort of a random process where cells are flowing into these emulsions and entering these droplets. And so at a low rate, you can actually have 2 cells enter the same droplet. And so we have ways of filtering out doublets. Doublets can be very misleading because if you do sequence this, all the mRNAs associated with the cell barcode, now you see transcripts that are coming from 2 different cell types. So you might see transcripts coming from a T cell and an epithelial cell, and you think you have something very interesting because now these T cell genes are being expressed in something that looks like an epithelial cell, but it's actually just a doublet. So you need to be really careful about filtering out these potential problems, so we have approaches for that. And then finally, we cluster all the cells together. So we take the 3 different tissues that we process from the same non-small cell lung cancer sample, and we cluster them all together, and we assign the cell type. So we see the major cell types. We see the malignant cells, which are these epithelial cells. We see immune cells, such as macrophages, mast cells, T cells, B cells, and we see stromal cells like endothelial cells and fibroblasts. And then we can ask here, what's the breakdown in terms of cellular composition across the 3 different protocols? And what we see is these last 2 protocols have sort of similar cellular compositions where these colors match the cell types you see here. The C4 protocol tends to capture more macrophages and more of the malignant epithelial cells. So which is better or worse? Well, it depends on your biological question. If your goal is to capture the most diversity of cells, maybe these last 2 protocols are really good. But if your goal is to really study the malignant cells, maybe you want a protocol that captures a bigger number of malignant cells. So those are things that we try to consider and point out when suggesting what protocols you should be using to process these cancers or these tumors. And then finally, one other really nice thing we can do with this data is we can infer the copy number variation. So within tumors, there's often large chromosomal amplifications or deletions. And so what we can do is for every single cell, we can look at the expression of genes, and we can look at very large tracts of genes. We can look at 100 genes at a time that are contiguous on the chromosome and compare their expression in a cell that we think is malignant to a cell that we think is non-malignant. And if we see in the tumor cells that, at chromosome 1, there's very high expression across hundreds of genes, but in non-malignant cells at chromosome 1, there's very low expression of genes there, we can say maybe there's been an amplification. And so here we have our malignant cells in orange, and we've inferred the copy number amplifications by comparing the gene expression in these chromosomal windows to the gene expression in this reference set of cells that we are very sure are not malignant cells. And we can see the different amplifications in red and deletions in blue. And we have validated this type of approach with whole exome sequencing. So we know that this is actually working, and this gives us, at a single cell level, information about some of the genetic architecture of those cells. And often, not in this example here, but in glioblastoma, for instance, we see different clones of the cancer. So within a patient, we might see that, there's amplification in chromosome 1, but in one set of cells, but not in another. So we see the different cancer clones. And so this is really nice, but we can get a single cell measure of the genetic architecture without actually doing any kind of genomic sequencing of the single cell. So this is also a really powerful approach for understanding what's going on the tumor side. Okay, and so we did this for many different cancers, and here's just showing some of the examples from lung cancer, breast cancer, ovarian, leukemias, neuroblastoma, and glioblastoma. Okay, and so the last part I'll talk about is single nucleus transcriptomics. So everything I've been talking about up till now is single cell transcriptomics, where we dissociate the tissue into single cells. However, it's often not possible to dissociate a sample very easily, or we are starting with a frozen sample or something that's been lightly fixed. There are many frozen samples in hospitals. When a tumor is collected, chunks are sliced out and put in a -80. And the question is, can you use those tumors, those samples, and learn anything? And you can't just do single cell sequencing on this, because if you thaw a frozen tissue, there's going to be incredible damage to the cells, to most of the cells. And so you can't really learn anything from that. But if instead of looking at all this mRNA that's in the cells, you could somehow extract the nuclei of those cells, the RNAs in the nuclei are much more protected because they're sitting in this organelle, and you don't have to dissociate the sample. So an approach that we started to develop 3 years ago is actually isolating the nuclei from the cells. And this has been really successful, especially in the brain, but we tried to apply it to tumors as well. And so the advantage of this is that, again, as I said, looking at the nuclei does not require tissue dissociation. There are many populations that are lost during normal cell dissociation. So epithelial cells, for instance, like to stick together, and when you dissociate them, often they just die or they lyse or something bad happens. But with nuclei, this isn't an issue. Another advantage is we can go back to banked samples. So many clinical diseases are quite rare. So childhood cancers, for instance, it's not like you could start a project where you wanted to collect 100 neuroblastoma samples. That would take you probably 10 years. However, there are 100 neuroblastoma samples in many freezers of hospitals that have been accrued over time. And so being able to access those samples and learn something from them can be very powerful. Another reason is, like I said before, there's very tight coordination that has to happen when you're doing single-cell RNA sequencing, meaning that you have to go from getting something out of the patient and delivering it to a pathologist and then having it processed immediately by a molecular biologist. This often takes a day. Sometimes we have molecular biologists who receive a tumor at like 8 p.m., and they are up at 4 a.m. like doing dissociations and processing, because everything has to be done fresh. So if you're able to freeze the sample very early on, that makes your life easier, and it also allows you to process many samples in parallel together, reducing batch effects. So then a question you might have, though, is, well, the nucleus is very different from the whole cell, so maybe the gene expression is just not very interesting in the nucleus, or just so different that it's not something we can use. So that's a good question, and I'll address that at the end. But what a postdoc in the lab did was they tried to develop sort of a toolbox of different techniques for isolating nuclei from tissues. And so they tried many different tissues, brain, lung, esophagus. In addition to tumors, they tried many different ways of isolating the nuclei, mortar and pestle, cutting up tissues, downsing, different enzymes and detergents. And they also tried different ways of isolating the nuclei, whether sorting into plates or doing droplet technologies. And so in the end, there were like 104 preparations that we tried, and we narrowed down on the preparations that worked the best. And then we can recommend the best working preparations to other people who are trying to get single nuclei sequencing to work in their lab. And so again, we developed this sort of decision tree of what to do with a frozen sample. And these are the 4 sort of best protocols that we decided on for dealing with frozen samples. And if somebody is going to, take a new tissue that's been frozen, this is what we recommend they start with. And again, we kind of evaluated the different protocols based on these 3 criteria: how good do the nuclei look, how many do we recover, what's the cellular composition. And so to give you a sense of like how well this worked, I do a lot of work on neuroblastoma. And so when I started on neuroblastoma, this is a typical, and neuroblastoma is a childhood cancer. This is a typical sample I would get where you see this sort of big blob of cells, not too many cells, maybe a few hundred, and you don't really see distinct cell types that you would expect in a tumor, namely tumor cells, immune cells, stromal cells. You just see this big blob. However, after doing these optimizations, in the last year or so, we start to see very distinct clusters. We see the neuroendocrine cells, which are the malignant cells. We see T cells and stromal cells and so forth. So all these optimizations really worked. And it was very exciting to see, because we went from basically saying, okay, we can never use these samples that are frozen in the freezer, to now we've processed close to 50 samples from neuroblastoma patients, and we're trying to understand some of the differences across neuroblastoma. And again, you could never really collect this many samples by, starting a study where you're enrolling patients. So one of the major differences when dealing with single nuclei versus single cells is that many of your mRNAs are pre-mRNAs, so they haven't been fully spliced and processed. So many of the reads are actually going to map to the introns. So when you do your sequencing, many reads map to introns. Those introns would eventually get spliced out in a mature mRNA, but many of your, like I say, much of your RNA is coming from these pre-spliced mRNAs. And so what we do is we actually use the information from the introns in our analysis, and we don't just throw that out. Okay, so here's an example of neuroblastoma where we tried 4 different protocols. And what we can see is actually for this case, the 4 different protocols were pretty similar, although these latter 2 protocols gave a little more diversity of the cells that weren't just the neuroendocrine cells. Okay, and then we were able to do this across many different cancers, and essentially for every cancer we've looked at so far, we've been able to apply one of these 4 protocols for frozen samples. And then finally, well, how much what's the comparison between doing something with single nucleus where you're collecting RNAs from the nucleus versus single cell where you're collecting RNAs mostly from the cytoplasm? So what we did was we took a neuroblastoma sample that came out of a patient and we freshly processed it, but we also froze a chunk of that sample and we did single nucleus processing of that. And so we have a matched sample where we have single cell and single nucleus coming from the same sample, so that cuts down on heterogeneity that you would see across tumors from different individuals, and we clustered those samples together with a data integration method. And you can see the different cell types here, and it forms this, in the visualization, it sort of looks like a poodle, like a small dog. And then we can, let's go back. And then we can look at just the cells alone or just the nuclei alone. And what you can see is that overall, the cells in the nuclei sort of overlay with each other pretty nicely. And we are identifying the same cell types. So we're finding, malignant cells or T cells here in both of these. We're finding neuroendocrine cells. We're finding, macrophages and so forth. So the same cell types are being identified across the single cell and the single nucleus data. However, as you can probably see by eye, the proportions are a little different. So within the single cell data, we have a bias towards recovering more immune cells. So this black bar is T cells and NK cells. And in the single nuclei data, we have a bias towards a more sort of even representation of all the cell types. In addition, what's interesting is we're better at recovering the neuroendocrine cells. So those are the malignant cells in neuroblastoma. And so if you're studying neuroblastoma and you really want to look at malignant cells, actually doing single nuclei is a better approach than taking freshly dissociated samples and processing them. And again, that's because the malignant cells are often sticking very closely together. They're often very hard to dissociate. But when you do single nuclei, you get past the whole tissue dissociation problem. So just to summarize that, sort of how do you choose the best protocol? Well, we often try to just take one single piece of tissue and compare a few protocols in parallel. These are protocols now that have already been pre-selected by us doing many, many comparisons, so other people don't have to do those comparisons. It's often good to compare doing single cell and single nucleus on the same tissue, because single nucleus can sometimes be better and have many other advantages other than just the data quality, but just the ease of getting samples. And then finally, like what's best depends on your biological question. If you need a protocol that recovers the greatest cellular diversity, that's one set of biological questions. If you really just care about malignant cells, you should be trying to choose a protocol that just gives you more malignant cells. And so it's not just like there's one best protocol that applies to that tumor or across many tumors. You really have to tailor things to the question in the tumor that you're looking at. Okay. So I'll just thank all the great people I work with, in particular Mario, Kai, Aviv, and Orit, and have a lot of acknowledgments. So these studies are like very big, as you can imagine, because we have many tumors and many doctors and pathologists and technicians and graduate students and so forth. So here's some more acknowledgments and some more acknowledgments. But most of all, the most important is the patients and their families who care about research and support it and donate these very precious samples. And with that, I'll take any questions you have. Thank you. Yeah, Mario.
It is very impressive. So you know that now it's getting popular to generate tumor organoids in order to make experiments in vitro and, for instance, to test different kind of drugs.
Yeah.
So somebody has been performing an experiment in which you take the tumor from the patient and then from that to create an organoid and then to make a single RNA-Seq analysis from the organoid and the original tumor and see how similar those are?
Yeah, people definitely, that's sort of the first kind of experimental control you want to do, which is you take the patient's tumor, you process that, but you also take some of the cells from the patient's tumor and grow up an organoid, and you do single cell RNA sequencing on both of them, and you compare the gene expression profiles and make sure that they're consistent with one another. It often is very tricky because you have to try many different conditions to grow the organoids, and these are, it's biology, it's messy, so you try a different cocktail of things in your media in order to make these things grow, but maybe that thing that you added, that cytokine or whatever, leads to changes in the expression profile. So you have to be very careful, and so you have to screen many different conditions, but exactly what you're saying, you have to compare it to the original tumor. But yeah, so sometimes this works very well, and it's possible to do organoids that match the original patient tumor, and sometimes it's very hard and not possible.
Okay.
Yeah.
So another question is if during your RNA-Seq analysis in different cancers that you analyzed, do you were able to identify cancer stem cells, and if these cancer stem cells are heterogeneous?
Oh, absolutely. Yeah, we definitely find cancer stem cells, especially in glioblastoma. Like I didn't show it here, but we have a lot of papers on that where you see the cancer stem cells, and you can see them sort of differentiating to one sort of cell type or another. And you can see it within the patient where you see a stem cell population where it's highly cycling, and then you see it going down one lineage or another. And so that's one cool thing about this type of approach, is that you can get that kind of lineage information because there's all these different states and types being sampled within the patient's tumor. So you can actually see where the stem cell is going towards, what state it's going towards. Yeah.
Thank you for the talk.
Sure.
I have two questions. One is related with the one that Mario just did. Is where I can find the differentiating cells or the stem cells within those spots that you show. Are within those spots or just are discarded from the analysis?
Oh, no, we don't discard anything from the analysis. Like, we're usually very careful to throw anything out. I mean, it's pretty easy to identify stem cells often because there's usually, we already know from other studies some of the gene signatures, so you can just take the genes you know are important and just look for their expression, and that will usually highlight it. Stem cells sometimes are cycling, they sometimes have markers like LGR5 or whatnot, so we can identify stem cells often pretty well. They also often are at the sort of kind of at the center of the cluster as they, and everything else is sort of related to them. So just where they're positioned in the clustering also is informative.
My other question is, I don't know if it's possible, is, I mean, the tumor is heterogeneous at the cell level, but also at the genetic level. My question is, is there any way to relate the genome and mutations within that genome and the transcriptome?
That's a great question. So that's one thing that people are really trying to do, is relate the genetics to the transcriptome. So, I mean, we can look at copy number aberration just from the transcriptome, but there are techniques specifically where you do the single cell RNA sequencing, but when you do the single cell RNA sequencing, you're actually getting a first strand cDNA that covers most of the transcript itself. So you can actually take that library that has the first strand cDNA, which is the full-length gene that's been being expressed, and you can deeply sequence that and call the genotypes. So there's this approach called like genotyping of transcriptomes, where if you start with the transcriptome and you have the first strand cDNA, and you call the mutations that are at least in the transcriptome. And that's actually not you don't have to do anything special to you just have to add an extra set of molecular biology steps, but there's no machinery or anything extra needed. So people are relating the genetic heterogeneity of the cancer to the transcriptome program and seeing to what extent there's a link. So sometimes, like the cells cluster by transcriptome, and then you look within those clusters, you see unique genotypes, and sometimes you see a mix where within a single cluster of cells, there's many different genotypes. So sometimes there's a link between the genotype and the transcriptome, and sometimes there's not a link. And when there's not a link, you start to look at things like epigenetics and approaches like that. Yeah. Oh, that's me. Very strange. Yeah. Yeah. Oh, I see.
How can we integrate single cells? So it says, how can we integrate single cell omic technologies to other data sources? I mean, other omics.
Okay, that's a good question. So, first of all, there's a lot of there's a move towards these multi-omic technologies where you can measure from the same cell, like we were just discussing, both the transcriptome and maybe the genotype, or sets of proteins, or chromatin accessibility. So from the same cell, you have multiple types of measurements, and there are analytical approaches that are being worked on to kind of integrate things like that. And a lot of the times, it's kind of like a dimensionality reduction problem where rather than just having one biological feature, you have multiple biological features, and you want to embed them in some lower dimensional space that relates the two things you've measured. So that's one thing, is if you're just doing single cell. But if you're not just doing single cell, if you have other data sets like bulk RNA sequencing, what you can do is take your single cell data where you have a signature measured, for instance, like we saw in this example for the melanoma and the T cells, you can get a signature for T cells and then look at the expression of that signature in your bulk data set as a way to sort of expand the number of data points you have per patient. So it's sort of right now that's one of the big sort of movements, is to sort of combine data sets together. So pretty much any approach you come up with will be novel at this point and worth trying. So there's no like right or wrong thing to do right now. I mean, there's probably some wrong things you could do, but a lot of correct things you could do too. Yeah.
¿Alguien más aquí? ¿No? ¿Sí? Yes.
My question is about when you make me think about when you said that with the microfluidics field in the droplet you can get 2 cells.
Yes.
And then you filter those data.
Yeah.
But then what happens if some of these cells are transdifferentiating?
Oh, that's a great question. Yeah.
For example, if there's epithelia to mesenchymal transition.
Yeah, that's a great question. So sometimes, a lot of the times when we see cell clusters that seem to have features of 2 different cell types, those are very interesting to us because that might be a very exciting transition, but it could also be that, like you're saying, it's a doublet where you have those 2 different cell types just by accident got in the same droplet. So first of all, the rate of doublets is often pretty low, but essentially it's always hard to prove that what you're looking at is not a doublet. So you need some external validation. So for instance, if you see an epithelial to mesenchymal transition and you see that these genes are being marked, then you would go and probably do like either immunofluorescence or immunositu hybridization to go and try to find the expression of those unique genes in a tissue slice and show that that population really exists in the starting tissue. It isn't just observed when you do the single cell dissociation. So it's kind of like anything in biology, you just need another validation that's independent of what you start with because it's very circular. You just can't escape the fact that it could always be a doublet. Yeah, that's very tricky.
That's interesting.
Yeah.
Thank you.
Yeah.
Yeah.
Oh.
Between using resection or biopsies, yeah. So there are differences in that certain protocols definitely work better for biopsies because the biopsies are often very, very small. So we have sets of instructions for dealing with the biopsies themselves because for those it's like very bad to lose cells because then we don't have enough cells to sequence. But yeah, that's described in the bioRxiv paper, like how to deal with really small samples like that. But yeah, there are things that we really consider. Resections are much easier.
Hi, here. Amazing story you have and a lot of data, and I'm very glad that you put all of this data available for the brave people who want to get through this data.
Yes, that's great. Yeah.
Just I'm wondering, our classification of the different cell types, especially with the immune cells, it was done somehow with other criteria that is much less sensitive than the total gene expression of the cells.
Absolutely. Yeah.
Do you think within your data we could make a better distinction to include more cell types?
Yeah.
For example, in the case of the B cells, I clearly saw 2 different B cells. I mean, not completely different, but they are not just one.
Yes.
Do you think you can reclassify the cell types with this data?
Absolutely. So one of the goals of the human cell atlas and any of these types of approaches is to really classify the cell types using more quantitative data because, as you know, in immunology, they've done amazing progress using surface proteins and flow cytometry and classifying cell types. But we know that even when you have a pure cell type, there's often still some heterogeneity there. And that's probably because there's multiple states within that cell type. So one of the biggest challenges is once you have a sort of cluster of T cells, how do you say what the states are? Because the states are a lot more continuous, like an exhausted T cell or a naive T cell. There's just a continual difference in gene expression between them. And so how do you say what's one state versus another? And so for there we go, instead of doing what's called like hard clustering, where I say this cell is 100% a T cell and this cell is 100% a B cell, I say instead that every cell is a mixture of states, where this T cell is maybe, has 90% exhaustion state, but also some naive state to it or something like that. And so that gets us away from trying to say a cell is one thing or another, because that's probably not the way to think about it, because we know there's this continual difference in the states. And yes, there are sometimes strong commitments made to one or the other, but moving away from thinking of it as a clustering problem, like a hard clustering problem, to more of a soft or fuzzy clustering problem is probably one of the conceptual changes you have to make in thinking about this. So thinking about it as every cell being a mixture of states rather than one thing or another is one of the approaches that we're thinking about and taking, but it's a very hard problem. And you have to work with immunologists and also convince them because they're the ultimate experts. Yeah.
And the other is, were you very surprised to find this large difference within the different individuals?
Yeah.
That they are not, I mean, I guess my expectations would be that they will cluster closer together. And
Yeah, it was very surprising to see that, but it also makes sense with the genetic heterogeneity of cancer. So it just underscores the challenge of any kind of treatment. But we do think that, yes, there is heterogeneity, but we do have approaches to still find some common gene programs, even when these things cluster separately. Like most of it might be different, but there's still, if things cluster separately, that doesn't mean they necessarily have nothing in common. It just means there's a lot of variation, but there still can be common programs. And so we try to look still for some commonalities that we can sort of target or think about. Yeah, but it's a really hard problem. Yeah. Yeah.
Me again, and it's kind of philosophic, but under the light of these new tools, and under your terms now, can you define us a malignant cell? What is a malignant cell when you consider it malignant?
Yeah, it's really hard. We try to use multiple criteria. So sometimes it's like known, for instance, in like a lung cancer that it's going to be something, a cell type that's going to be somewhat epithelial. We try to look for these like copy number aberration events. We try to look for lots of cell cycling. It's usually multiple different biological signals that we look for in the data. Yeah. Yeah, well, but because we're looking at every single cell individually, we don't need to worry as much about like confusing a malignant cell for a T cell. But sometimes it can be hard to tell because, you know, in the tissue of origin too, there might be some tissue that's not transformed, and so it might look like a malignant cell. So it's a hard problem, and it's just like you have to take it on a case-by-case basis. And again, you try to use other pieces of data that aren't just from your single cell study.
Yeah, but for example, when you say that the black ones are the malignant cells because they are fulfilling 5 criteria, 10 criteria, how many?
I mean, we usually have like 3 criteria, but you have to be careful and, you know, especially for something where there's less known about what's going on. Like we're studying cancers where at least glioblastoma is heavily studied, neuroblastoma less so. So we expect to find more surprises in neuroblastoma because we don't have much prior knowledge. So we are really careful and try to keep our kind of mind open as to what we're seeing and whether it's really malignant or whether it's just a weird acting chromaffin cell that's changed because it's in this environment. Yeah, but it's a hard question. Yeah, and I think you have to ultimately validate with some other technique. Yeah. Yeah.
I have one question. A very nice talk, very impressive amount of data. Regarding your last comment about the heterogeneity in all these tumors from among people, does that reflect on, can you find a common signature in the T cells that is affected by all this heterogeneity that makes the T cell not being able to control the tumor?
We did try to look at the T cells themselves. Like in that study with the melanoma cells and the T cells, we didn't just try to make signatures in the malignant cells that were predictive. We also tried to make signatures in the T cells that were predictive. In a way, that's like another, like kind of, you know, the other side of the coin. But it wasn't very predictive. Part of that might be because I think sometimes the signal gets a little diluted with the immune cells in the single cell tumor data sets, because what happens is that when you resect a tumor, it's a very messy process, and there are blood vessels and normal blood at the site of the tumor are very close, and so it's hard to make sure that that you don't get any like just sort of healthier blood in the sample, and that gets mixed up with the T cells that are actually at the site of the tumor. So that can dilute out the signal, I think. That's like one hypothesis we have. And just from talking to like pathologists and surgeons, they say it's they always see blood that's not from the tumor there as they're cutting it out. So like they see that they cut a blood vessel or something, you know, it's not a perfectly controlled sample. So we can't really say it's purely tumor, everything. But we know the malignant cells are purely tumor, but the T cells, some of them might be from healthy blood, and that probably dilutes the signal a little bit. But we certainly look for signatures in the T cells, absolutely. And
Thank you.
Yeah, sure.
¿Alguna más?
Okay, thank you very much.